Okay, so we're going to be going on to the second part here of Chapter 12, Part 2. We're going to be continuing on. Again, these are going to be scenarios which do not follow normal Mendelian genetics. Okay, so we talked about a bunch already. We're going to talk about some more here. So the first is pleiotropy. So when Mendel did his studies, one gene controlled one trait, right? So the flower color gene controlled flower color. Well, that's not the case with every gene. Sometimes one gene has multiple phenotypic effects. So instead of affecting one phenotype, it actually affects multiple phenotypes. Okay, so again, that's called pleiotropy. One gene, multiple phenotypic effects. Some examples of pleiotrophic alleles. There's some of these examples here that I have listed for us are recessive hereditary diseases. So for example, cystic fibrosis. So cystic fibrosis is a recessive hereditary disease. Individuals who have cystic fibrosis must have inherited two recessive alleles, one from each parent, and therefore they express this recessive disease, cystic fibrosis, which is a pretty nasty disease. This one gene affects multiple organ systems. So there's one protein that is defective, and that one protein is going to wreak havoc on multiple organs and organ systems throughout the body. People with cystic fibrosis, because they lack this protein or they have a defective protein, they end up building up thick kind of viscous mucus in all sorts of organs. So their lungs, their digestive system. In their lungs, it's going to impair breathing and gas exchange. In their digestive system, it's going to impair digestion. Okay. Another example is sickle cell disease, and let's talk about this for just a second. So this is pretty interesting. So for those of you who took General Biology 1 with me, way back in Chapter 3, I think, when we talked about protein structure, we actually looked at sickle cell disease, if you can remember back that far. I know it was a long time ago, so let me remind you. So oxygen is carried in your red blood cells by a protein called hemoglobin. Okay, and hemoglobin is actually made up of 4 polypeptide chains, 2 alpha chains and 2 beta chains. People with sickle cell disease, they actually have a genetic mutation. So they've got the recessive allele that has a mutation in it, and that mutation it's a single substitution in the protein. And so people who have sickle cell disease, their beta polypeptide chains, because of that single amino acid substitution, the polypeptide chain doesn't fold properly. Okay, so remember, form equals function when we're talking about proteins. And so since the beta chain doesn't fold properly, it doesn't have the right shape, and therefore it can't bind to oxygen very well. And actually, the hemoglobin molecules tend to actually stick together and form these chains, which gives red blood cells a crescent shape instead of being nice and concave and kind of circular. So in sickle cell disease, this is a recessive hereditary disease, and individuals who have this disease have a faulty protein. And that faulty protein causes a misshaping, a sickle shaping of their red blood cells. And so this has multiple effects. So red blood cells are not as good at carrying oxygen as normally shaped red blood cells. So there's one thing. And because of that, all of the cells and tissues in the body are going to be kind of oxygen deprived. So there's something else. And then because of the sickle shape, the kind of crescent shape that the red blood cells end up taking on, those red blood cells tend to get stuck in capillary beds, and therefore they cause blood clotting. So again, one gene having multiple phenotypic effects throughout the body. Okay, again, that's called pleiotropy. Okay, next slide. Now the opposite can be true as well. So in pleiotropy, you have one gene, multiple phenotypes. You could also have multiple genes contributing to one phenotype, which is the opposite. Okay, so again, not every characteristic is controlled by a single gene. many characteristics or traits are controlled by multiple genes, and those genes may interact in some way. Here's an example. Now, I don't know very much about chickens, but I like this example because it's a visual, and I've got some more on the next slide. Okay, so the comb of chickens is going to be controlled by two genes. We've got the R gene and the P gene. Okay, and these two genes, depending on the combination of alleles, we can end up with 4 potential different phenotypes. So this first chicken here is displaying the walnut comb. Okay, pretty neat. And this is going to be coded for by any of these genotypes. So if the chicken is homozygous dominant for both the P and R gene, it will have the walnut comb. If it's big P, big R, walnut comb, big P, big R, or big P, big R, little r. Any of those genotypes will give you the phenotype walnut comb. And actually, I just want to make sure that we understand this really quick. We could have did all of this shorthand, and we could have said any chicken who is big P dash, big R dash, that should be a dash here, is going to have the walnut comb. What that tells us is that it doesn't matter what the second P or R allele is, whether it's dominant or recessive, because the chicken will have the walnut comb. Okay? The P comb, the same thing, we can do some shorthand here. We could say, gotta find a free spot here, big P dash little r is going to give you the P comb. Okay, so if it's big P or big P, little r, both of those will give us this phenotype of the P comb. The rose comb, which looks like this on this chicken, is coded for by little p, big R dash. Okay, so big R, or big R will give us that phenotype. And the last phenotype here, which is probably my favorite, is called the single comb. Looks kind of like a mohawk. That's going to be the homozygous recessive genotype. Okay, so again, we have two genes here that are contributing to one phenotype. Okay. Move my picture here. You know what? I may have to minimize this. Maybe I can put it up there at the top. That's fine. Okay, so we're still talking about multiple genes, one phenotype, but in this case, this is when one gene, one genetic locus, controls the expression of a second gene. Okay, when that happens, we use the term epistasis. Do I have my pen? Yeah. See that term right there, epistasis. And you actually will see this week in lab as well. So when you get to corn cob 4, which is activity 2, case 3 in lab, you're going to have a corn cob that shows a 13:3 ratio of yellow to purple kernels. And in that particular case, there's going to be a gene called the C1 gene. So either the C1 or the C, those are the two possible alleles for that particular gene. And the C1 is called the color inhibitor allele. So if C1 is present, there will not be any color in the alluron of the corn, and so the kernel will appear yellow. Okay, so again, you're going to get that in lab. So that C1 gene controls the expression of the color gene. So that's called epistasis. We would say that that C1 gene is epistatic to the color gene. Here's some more examples of epistasis. In this particular example here, we've got some mice. Okay, and so in these mice, let's see. The mice can be brown, that's the wild type color, and that can be coded for by big A, or big A, little a. The recessive color is going to be little a, which is going to code for black. Okay, so these mice can either be brown or black. However, there is a second gene that controls the expression of the A gene, and it's called the C gene. In this case, the capital, the dominant, so the big C, is required for pigment production. So now this is not inhibition like you're going to see in lab. Instead, this is required in order to have pigment. So there has to be a big C allele present to get black or brown. If it's little c, the mice will be white. Okay, so let me write this really quick. So little c, dash, dash. Doesn't matter what's there. It could be big A, it could be big A, or it could be little a. In any of those three cases, we're going to get white mice. Okay. The only way to get a brown mouse is big C dash, big A dash. And the only way to get a black mouse is big C dash, little a. Okay, actually I wrote the reverse of how the Punnett square is. So it doesn't matter which gene you put first, the A gene or the C gene, as long as you keep the A's together and the C's together. Okay, so let's take a peek here at this cross. So we've got two brown mice, and both of their genotypes, they're both heterozygote at both loci. So they're both big A, big C, little c. When we set up this Punnett square, we're going to do it just like we did for the dihybrids. Okay, so you're going to do that FOIL method. Okay, so potential gametes for this mouse, for this mouse are going to go on this side, and then gametes for this mouse are going to go here. So we could have, I think it's too small for me to actually write the lines here, so let me try this. Let's see. Okay, so for this mouse, here's its genotype. So you're going to go here and say, okay, so 1/4 of the gametes are big A, big C. Here, 1/4, oh, they did it in the reverse. So our big A, little c, 1/4 our little a, big C, and then 1/4 our little a, little c. And it doesn't matter the order of your gametes. They can be in any order. I just like to go from A and go over, and then little a and go over. Okay. The same thing for this parent. It has the same genotype, so the same 4 gametes will be produced, 1/4 chance of each. And then inside your Punnett square, you know how to do this, right? You're going to fill it in. So if these two come together here, you end up with that genotype. Big A, big C, big C. Now, it's going to be a brown mouse. Why? Because the dominant form of the C gene is there. Okay, so we get color deposition, and we have a big A allele. And so it's going to be a brown mouse. Okay, this is going to be a brown mouse. This is going to be a brown mouse. Brown mouse, brown mouse. Okay, how do we get black? So yes, there's a color deposition gene, so we can get color. Now, what color? Well, this mouse is little a, which we know codes for black. Okay, so any of these that you have a big C with little a, that's going to be a black mouse. And any of them, if you have a big C and a dominant form of that A gene, you're going to end up with a brown mouse. Now, these albino mice, doesn't matter what's there for the A gene, because they're both, all 4 of them are going to have that little c. And so there is no color deposition. There's no pigment. So these mice can't be brown, they can't be black, they're going to be white or albino. Okay? This ratio here, 9:3:4, holds true if the epistatic gene is required for the trait to be expressed. You're going to see the opposite of that in lab. Again, you're going to see the 13:3 ratio, and that's when the dominant form of the gene, of the epistatic gene, inhibits the trait from being expressed. Here's another example of epistasis. And if you look here at the bottom, let me move this over. Look at, there's this ratio again, 9:3:4. Okay, same ratio that we saw on the past slide. On this particular one, my favorite animal, Labradors. So, Labradors can either be black, brown, or yellow. Okay, so again, there's a color deposition gene. So in this case, it's the E gene. So in order to be brown, in order to be brown or black, there has to be at least one dose of the big E gene. It doesn't need to be 2, but there needs to be at least 1. So any dog that is little e for the E gene, doesn't matter what's there for that second gene, it's not going to have any color. Okay? Now brown is recessive to black, so this brown dog will be little b, big E dash. Could be big E or big E little e. And the black lab is going to be big B dash, big E dash, which means that that dog can be big B, big B, big B, sorry, big B, big E, or big B, little b, big E, or that second allele for the letter E could also be big or little. Doesn't matter. Okay? So for a black lab, you have to have 1 dose of the dominant form of the B gene plus 1 dose of the dominant form of the E gene. Okay? For brown labs, it has to be homozygous recessive for the B gene, and then can be whatever, either heterozygous or homozygous dominant for the E gene. And again, anytime you have little e, you're going to end up with a brown dog. So at the top here, if we mate 2 heterozygous black labs, that's what this Punnett square is representing here. Okay, so again, gametes of the female dog are down this side, gametes of the male dog are across the top. Okay, and inside the boxes are potential offspring. So if this egg is fertilized by this sperm, this is what you get. So you can see that all the black dogs have at least 1 dose of the big B and 1 dose of the big E. Okay, so all these black dogs here. The brown dogs are going to be little b, big E, whatever. Big E, little e, doesn't matter. They're going to be brown dogs. And any dog here that has 2 doses of the recessive E gene is not going to have any pigment deposition. So their fur is going to be, albino, which is yellow. Okay? So again, another example of epistasis. In this case, the E gene is controlling the expression of the B gene. Okay, so on to linked genes. So these are not X-linked genes. That's different, right? We already covered that. These are linked genes. When you hear that term linked genes, that means that these are genes that are found on the same chromosome. Okay? Which means, gene A and gene B, coding for 2 different traits, they're located on the same chromosome. So that's important. If we have genes that are found on the same chromosome, they're not going to sort independently. Okay, so you got to bring yourselves back to meiosis 1, to metaphase 1, when homologous chromosomes line up on the metaphase plate. We said that homologous chromosomes, as they line up in their pairs, they're going to line up independently of each other, randomly. And that would give us those different genetic combinations that we saw. So if I go back 1 slide, maybe. Okay, let me get rid of some of this. Put this up here for a second, and let me get rid of all the ink on here. Okay, so if you look here, we had our 2 dogs, right? And we said that these are the potential gametes of one of these dogs. Well, that's because the B and the E gene were on separate chromosomes, and so they segregated or separated independently of each other during meiosis, and so you end up with all these 4 potential gametes. But what if the E and the B gene were on the same chromosome? Then they wouldn't sort independently, okay? Which would mean this looks different. You wouldn't end up with 4 gametes necessarily, not necessarily, okay? Unless you get recombination, which we're going to talk about. Okay, so linked genes, which are genes found on the same chromosome, they're going to behave differently than what we've been used to. They're not going to follow Mendel's second rule, right here, the law of independent assortment. And instead, they're going to be inherited together, unless crossing over and recombination occurs. And that's why I made you, in chapter 1 and in that first lab, demonstrate what crossing over is and how that changes the outcome in gametes. That's really, really important. Let me give you the history behind how we figured this out. I always like to give credit to those scientists who did this work. Okay, so again, it was Thomas Hunt Morgan's lab, the same guy that we talked about when we looked at X-linked traits, flies, the eye color. Same lab figured some of this stuff out, along with his students. Okay, all of his graduate students that were working in there really helped him to figure this out. They demonstrated this phenomenon that we call linkage. Okay, and again, what that says is that genes are on the same chromosome. And very interestingly, Morgan said, well, if they're on the same chromosome, can we figure out their order? And we could. So he was able to show genes that are linked, arranged in a linear order. Again, his model organism was the fruit fly. Fruit fly is a great model system because they're small, they're easy to house. You don't need a big animal facility to keep them. All you need is a little vial and you can set up crosses. They have a relatively short life cycle. They only have 4 pairs of chromosomes, so genetics is really easy to study. And I'm going to give you kind of the summary of their work. So what they did is they performed a 2-point test cross. Okay, so remember, what does that mean? He's going to take heterozygous flies, okay, flies that are heterozygous at both loci that he's studying, and he's going to cross them with flies that are homozygous recessive for both loci that he's studying. So this is really important. Whenever you see, an organism was test crossed, you always want to go to, okay, test cross means I need to mate this, whatever it is, with a homozygous recessive. Okay. There will be some questions in the genetics problems as well as in the lab book that are going to ask you to test cross. Okay, so make sure you know what that means. The traits that he was studying when he did these studies were body color and wing size. Okay, so body color, wing size. Those are the 2 traits that he's going to follow. Okay, and so for body color, let's look at that first. Grab my pen here. So flies could either be gray or black. And in this case, gray is dominant to black. And so gray flies would be produced by either big B or big B, little b. Black flies are going to be recessive. Okay, so black is going to be little b only. And then we've got wing size. So wings can either be normal or long. And the common terminology to use here is the letter V. Okay, and again, when you're doing these genetics problems, you can pick any letter that you want. Okay, you don't have to stick to the common way to do it. So normal wings would be coded for by big V or big V, little v. That's going to give you normal long wings. Where short wings, which are called vestigial wings, truncated, short, was the mutant form, okay, or non-wild type form, are produced by little v, so the homozygous recessive. Okay, now we don't have anything else going on here. There's no epistasis, there's no pleiotrophy, it's not X-linked, none of that. These are just genes that are found in the same chromosome. Okay, so we're looking just at that. So again, we've got body color and we've got wing size. And so Morgan's lab started to see some kind of weird things. So they said, okay, let's do a test cross here. We're going to take flies that are going to be homozygous. Let me erase that. So we've got parents that are technology here. So we start off these studies by crossing 2 homozygous parents. So the parental generation, the P generation, would be one fly that is homozygous dominant for both the B and V gene, and the second fly is going to be homozygous recessive for both the B and V gene. And so we cross those flies and we get the F1 heterozygotes, which are all big B, little b, big V, little v. Okay, so all of these flies here, they happen to be females. Again, this is not sex-linked, so don't get confused there. All of these females, the F1 females, are gray-bodied with normal wings, and they're all heterozygous. Morgan then test crossed these flies. So what does that mean? They're going to cross those flies with flies that are homozygous recessive for both genes. So little b, little b, little v, little v. Okay, and so now we just do our regular Punnett square here. And so Dad here only has the little b and the little v alleles, and so all of his sperm cells will be little b, little v. And then across the top, you want to do all 4 potential gametes. Okay, they line them up in a specific way here. So we've got big B, big V. We've got big B, little v. We've got little b, big V. And we've got little b, little v. So we've got all 4 potential types there. So now we do these studies. We look at the resulting offspring. Okay, now the offspring are listed here. So flies resulting from this fertilization would be gray with normal wings. Flies here would be black with vestigial wings. Flies here would be gray with vestigial wings. And flies here would be black with normal wings. Now, before we go on to look at any numbers, let me ask you, which one of these, or which ones of these phenotypes and genotypes are parental, and which ones are not parental? And so hopefully you would tell me that, well, mom is gray normal and dad is black vestigial, but these two look like neither parent. So they are new, okay? And in this case, let's look at the numbers. So normally, if there was no linkage, if these genes were found on different chromosomes and they sorted individually of each other, independently of each other, you would expect a 1-to-1 to 1-to-1 ratio. Okay, so here for expected results, these numbers here, if we had our total number of flies, we expect 1/4, which is 317 flies, to be gray normal, and 1/4 to be black vestigial, and 1/4 to be gray vestigial, and 1/4 to be black normal. So you'd expect that 1-to-1 to 1-to-1 ratio. The actual results here are not a 1-to-1 to 1-to-1 ratio. So the results that Morgan got looked like this. So if you look at these numbers, there's a whole lot more of these, the flies that look like one of the parents, and there's there's very few of these, the flies that didn't look like either parent. And so he hemmed and hawed and he tried to figure this out. And what he said is that, well, these two genes, the genes that code for color and wing size, must be linked. They must be found on the same chromosome, and therefore they don't sort independently of each other during meiosis. So then the question comes, well, then how the heck do you get any gray vestigial and any black normal flies? And the response there was, those must be the result of crossover and recombination. So these gametes right here are the result of recombination. So we call those the recombinant type. Okay? Now with this data, you could actually figure out the distance on the chromosome between the B and V gene. Okay, but before we do that, let's look back at meiosis. Let's make sure that we have it. Okay, so let's make sure we understand this whole parental, non-parental. So if we come back here, if we come here, okay, this is the cell of the heterozygous parent. Okay, so big V, little v, big B, little b. So that's, if we go back a slide, that's this parent right here. Okay, so gray, normal wings would be the phenotype, and the genotype would be big B, little v, big B, little b. Now, if we go to meiosis 1, inside of that organism's gamete-producing structures, okay, so we're inside of a cell that's about to make gametes. Let's follow what happens during meiosis. So during meiosis 1, we've got our homologous chromosomes that pair up to form a tetrad. So right here, this first figure, that's a tetrad, right? So a four-armed chromosome, it's going to come together. And if crossing over occurs, if there's a genetic exchange, right here, the little b and big B alleles are going to get swapped. And so we end up with the result here of meiosis 1. So now we have two cells. Here's one cell here and one cell here. We have got meiosis 2 that's going to occur. And look how we get 4 different gametes. We get the parental type, which would be big V, big B. No crossing over happened there. And the same thing here, this parental type, little b, which means no crossing over happened there as well. But here, these two in the middle, they're the result of crossing over. So big V, little b was not present in the original cell. And the little v, big B chromosome was not present in the original cell. And so those are called the recombinant types. Okay? And so if we go back here, again, these are the recombinant types shown up here. And recombination is going to be a chance event, a random event. We can actually calculate how often it occurs based on the numbers of resulting offspring. So let's actually go there next. Okay. We can calculate, let me move my picture here. We can calculate how often recombination occurs for a specific gene pair. Okay, so how often does recombination occur for the B and V genes? Okay, and then from that number, we're going to actually be able to tell the distance between those two genes. So this calculation is called the recombination frequency. What you're going to do here, so the formula is right here. You're going to take the number of recombinant offspring, and you're going to divide that by the total number of offspring. And then if you want a percentage, you're going to multiply by 100. So recombination happens what percent of the time? So let's use our example that we just went over. So here's the numbers that we actually got. Okay, number of offspring. So we're going to take the number of recombinant offspring and add those together. Okay, so 111 plus 106 is going to give you 217. That's going to be your numerator. That's the number of recombinant offspring. And now you're going to divide that by the total of all of the offspring altogether. Okay, which is going to be 1268. So you do that math, and then you multiply your answer by 100, and you should get 17%. Okay, 0.17, which is 17%. So the B and V genes, and these fruit flies, recombine about 17% of the time. Okay, it's called the frequency of recombination. Now, interestingly, we can take that number, so in that case it was 17%, and what we could say is that the B and V genes are 17 map units apart. So 1% recombination equals 1 map unit. A map unit is the distance on a chromosome. Okay? So from that, we can create a linkage map. So if you have a couple of different genes and you know the recombination frequency between each of those genes, you can figure out the linear order of those genes on a chromosome. That's called a linkage map. Okay? So in this little example here, and we've got some more in the genetics problems that we're going to be doing, the recombination frequency between the A and B gene is 5%, or 5 map units. The recombination frequency between the B and C gene is 3 map units, or 3%. So my question for you is, what's the recombination frequency between the A and C gene? We would know that it's 8. Okay, 8 map units. Okay, and then you can add in another gene. You can figure out what's the recombination frequency between the C gene in that gene, the B gene in that gene, the A gene in that gene. And then you can figure out where does that gene fit in relation to all 3 of those genes. Okay, so again, you can figure out the linear order of genes on a chromosome if you know the recombination frequency between those genes. Okay, so time for some questions. So you're going to go ahead and you're going to pause this here, take out your Are You With Me? And we've got this slide here, which is some definitions, and then we've got a bunch of genetics problems to do. So what I'd like you to do is take a stab at them, okay? And again, I'm going to go through them with you. Okay, so when you think you've got it, hit play and we'll work through it together. If you get stuck, try to go on to the next one. If you can't figure out one, maybe you can get the next one. Okay, and when you're finished, again, come back and we'll do them together. Okay, so let's look. Hopefully you worked through this one. So again, the big thing about genetics problems, they're word problems. And so they are tricky, and you have to keep really good record of what you got going on. So what I did is I've got this slide, and then my next slide over, if I can click to it, is going to be blank. So I'm going to go ahead and I'm going to work on this slide, but I'm going to have to keep going back and forth as I'm reading. Okay, so it says a dominant gene A causes yellow color in rats. Okay, so I would start off and I would write on my sheet of paper, I would put rats. That's the organism that I'm studying. And I know that big A, I'm going to grab my pen, hold on. So big A, we know equals yellow. That's what we know so far. Okay, let's go back. The dominant allele of another independent gene R, okay, so I've got two genes, one phenotype. I'm looking at coat color, and I've got two genes controlling this, okay? So this is a case of multiple genes. So, okay, so this R gene, or the R allele, the dominant allele, produces black. Okay, so big R equals black. All right. Okay. When there are two dominants together, they make gray. Okay, so big A dash, big R dash equals gray. Okay. So just from this, I know a little bit more up here at the top. So really, in order to get yellow, I got to have big A dash, little r, because if it was a big R, I would get gray. Okay, so I know more about that genotype. And for black, I also know more. It must be little a, big R dash. Okay, because if it was big A, if there was a big A there, it would give me gray. Okay, so that's how much I know so far. And then it says, rats of the genotype little a, little r are cream. Okay. Little a, little r equals cream. Okay. These genes are not X-linked. Okay, so all we really have going on here are multiple genes. So two genes affecting one phenotype, which is color. And we see that we get multiple phenotypes depending on the combination of those genes. Okay, so here we go. Here's the offspring. The mate, the cross. If a gray male and yellow female when mated. Okay, so hold on. A gray male and a yellow female. So a gray male. I don't know yet what he is. Oop, hold on. Much easier on a whiteboard. So big A dash, big R dash gives me gray. Okay, so now I'm going to mate that rat with a yellow female. So big A dash, little r. Okay. What am I going to get out of this? Okay, I'm going to get offspring, and I'm not going to be concerned about the numbers here. I put the numbers in, but you don't have to be concerned about them. So 3/8 of them are yellow. Okay, so I get some yellow offspring. So here's my parents. Okay, I want to just separate this stuff. And I'm going to throw my picture up here. So my yellow children are going to be big A dash, little r. Okay, do I know anything else yet? No. Okay. Then I get 1/8 gray. Okay, so I'm going to do a comma there. So my gray rats are big A dash big R, big R dash for now. Now, wait a minute. So if we look at mom, for the R gene, she only has recessive alleles to give. And so I know a little bit more about these gray rats. They must be big R, little r, because mom only has the little r allele to give. Okay, so there I just solved one allele there. Okay, 1/8 of them are cream. Okay, so cream is this. Little a, little r, little r. Oh, that tells me so much more. If I go back to my parents, okay, first off, let's look at the A gene. They both must have the recessive form of that A gene in order to get this. So I can come up here and I can fill this in. That's got to be a little a, and that's got to be a little a. I also know that for dad, that second R allele must be a lowercase little letter r, because the cream-colored children inherit one allele from each parent. And so this must be that. There, I think I answered the question now, right? So dad must be big A, big R, little r, and mom must be big A, little r, little r. Okay? Then there was some black children, black rats as well. And so the black rats are going to be little a, big R, little r. Oh, big A, sorry. Big A, big R, dash. We don't necessarily know that second allele. Okay, so there, I solved it. These are the parents. So yes, it was wordy and tricky, but if you do it step by step, you can hopefully figure it out. Okay, that one was a tricky one. Let's go to the next one. This one's pretty tricky too. Can you tell that I like genetics problems? They're fun. They're challenging sometimes, but they're fun to do. Okay, so now we're talking about the ability to hear. Okay, and this is going to be a case of, hopefully you know, epistasis. Okay. So we've got two genes here. I've got a fly down here too. We've got two genes. We've got the K gene and we've got the M gene. So there's two genes involved here. Multiple genes. And also it's a case of epistasis because of the M gene. So let's go ahead and read it. So it says, if the dominant K gene is necessary for hearing, so let's write this. So we got capital letter K equals can hear. And the dominant gene M results in deafness. Okay. No matter what the other gene is. What percentage of the offspring produced by the cross little k, big M, little m, crossed with big K, little m, will be deaf? It's kind of hard to tell with those K's what's capital and lowercase, so hopefully you got that. Okay, so first off, before we go and answer and do the Punnett square, let's think about the possibilities here. So if we write this, in order to be able to hear, an individual must be big K dash little m, That's the only way that you're going to get hearing. So it could be big K, or big K, little k, but it has to be little m in order to have hearing. Any individual that has this genotype, dash, big M, dash, is going to be deaf. Doesn't matter what's there for the K gene or the second M allele, that dominant big M is going to cause deafness. Okay, so let me erase this. And let's go ahead and do the cross. So we've got little, why isn't it working? Okay, hold on. We've got little k, I'm going to draw lines over them, big M, little m, mating with big K, little little m, little m. Okay, so let's set up our Punnett square. So we'll do this parent along the left-hand side. Okay, so the gametes produced in this parent are going to be little k, big M, and the other half are going to be little k, little m, and that's all. Okay, so it's going to be a smaller Punnett square. Let's see about this second parent across the top. So that parent's going to make big K, little m gametes, or half of them will be little k, little m. That's all I have. That's all that parent has. So we don't need to list any more. Okay, we could have repeated ourselves, but we don't have to. Okay, so inside the box, I'm going to actually grab a different color so that we can see the difference here. Let me grab black. So we've got big K, little k, big M, little m. Okay, this child's going to be deaf. Just that big M allele being there, we know it's going to be deaf. Second child, little k, oops, sorry. Big M, little m. That's just my dog. Again, deaf. Okay, third one here, big K, ooh, okay, little k, little m, little m. Oh, this individual should be able to hear. Okay, and the last one, little k, little m, little m. Okay, so remember, one dose of the capital, the dominant form of the K gene, is required to be able to hear. So again, this individual is also going to be deaf. Okay, and that's due to the K gene. Okay, there's a child screaming outside. Sorry, it's not mine. Okay, I closed some windows, so we shouldn't hear any more screaming. Maybe dog barking, but not screaming. Okay, let's go to the next one. So let me just finish, actually, I forgot to say this. So it says what percentage. So you want to make sure that you actually answer the question. So you would say, oh, whenever I pause it, it does that. You would say 75% will be deaf. Okay? And when you do these genetics problems in either lecture or lab, when you're doing them, you want to make sure that you really somehow designate what your answer is. So in this case, you would maybe circle or box in the 75%. That way, when I'm grading them, if you get the right answer, I can just kind of say, okay, it's good, you're fine. If you get the wrong answer, I can go back and I can look at your work and let you know where you went wrong. Okay? Okay, so next question is a long one. Oh. And so again, on this one, I'm going to read it with us, and then I've got a blank page here, maybe. Let's see. The blank page should be next, so that we can do our work on this one. Okay, so let's read it. So we've got Drosophila melanogaster, that should be italicized too, that's a scientific name. Fruit fly. Okay, that's what we're talking about. There is a dominant gene for gray body color and another dominant gene for normal wing size. So you could either do what we did on the past problem that we went through with this, with Morgan's flies, or you could pick a different letter if you want. I'm going to keep it the same. So I'm going to say that, oh, big B equals gray and little b equals black. Okay, this is going to be a case of linked genes that are found in the same chromosome. We have a second set of genes that code for wing color. Okay, oh, I already gave it away. So they are linked, but you can tell by the numbers here. We'll get there in a second. Okay, sorry, phone's ringing, dogs barking, kids screaming. It's that kind of day. So where were we? So we got two genes. We've got the body color gene and the wing shape gene. Okay, so for body color, gray is dominant to black. And for wing shape, normal is going to be big V, or long wings. And the recessive form, the V, equals short or vestigial wings. Okay, all right. So we take these flies and we're going to cross them, right? Okay, let me erase this stuff real quick. And let's see. So we're going to take flies that are homozygous for gray body color and normal wings. Okay, so if they're homozygous for gray, they must be big B, and normal wings would be big V, big V. And then we cross them with flies that had black bodies and vestigial wings. Okay, so crossed with little b, little v, little v. Okay, the only way to get black and vestigial wings is if it's homozygous recessive. And so the F1 progeny, right, this would be the parents. These are the F1s now that I'm going to write. They're all going to be heterozygous. So big B, big V, little v. Okay, so now it says to take the F1 progeny and then test cross them. So we're going to take these flies here and we're going to cross them with little b, little v, little v flies, right? Because that term test cross means to cross with homozygous recessive. Here's the results. Okay, what would you predict if this followed normal Mendelian rules and if these genes sorted independently of each other, if these genes weren't linked? Hopefully you would look at that and say, well, I've got a heterozygote and a homozygous recessive. That's going to be the 1:1:1:1 ratio. So a quarter this phenotype, a quarter this one, a quarter that one, and a quarter that one. Well, we don't get that. Instead, we get these numbers here. We get 236 flies that are gray normal, look like this parent. We have 253 flies that are black vestigial. Oh, they look like this parent. But then we get 50 flies that are gray with vestigial, which look like neither parent. That's a recombinant phenotype. And we get 61 flies that are black with normal wings. So again, another recombinant phenotype. So the question is, do you think these two genes are linked? Hopefully you said yes. And if so, how many map units apart are they on the chromosome? So how do I know that they're linked? By the numbers. So if I have a whole lot of parental and very few or much less of the others, non-parental, I'm going to know that recombination happened. So these genes must have been linked. Okay. If I got all the same, so if I had, I don't know, maybe 175 of each phenotype, then I would say no, the genes are not linked. And the evidence there would be because I have an equal number of each phenotype in the offspring. That would tell me that they weren't linked. But since I don't have that, I know that they're linked. I have more parental than non-parental. So let me erase this stuff. And I don't actually think I need the next slide. I think I can do this right on here. So how am I going to figure out how many map units apart? I need to figure out the recombination frequency. Remember that 1% recombination equals 1 map unit. I need map units. So how do I get percent recombination? I'm going to do the number of recombinant flies. So that's going to be 50. Oh, that's going to be 50 plus 61, right? These are the recombinant flies, divided by the total number of flies. So everybody. So that's going to be 236 plus 253 plus 50 plus 60. Okay, so if I did my math right, I did it over here. So if I add up 50 plus 61 and divide it by the total, you should have gotten 0.185. And now I got to multiply that by 100 to get a percent. So 18.5%. And remember that 1% recombination equals 1 map unit. And so you would tell me that these two genes, the body color and the wing shape gene, are 18.5 map units apart. Okay? Okay, on to the next one. This is the last one, I think. Okay, so this is a linkage map. So can we figure out the linear order of genes? So to start one of these problems, you're literally going to draw a straight line. Okay? You could do it vertically. I like to do it horizontally myself. And you're going to start to place the genes on there. So the first set of genes that we talk about are the A and B genes, and those are 40% apart, or 40 map units apart. They have a recombination frequency of 40%, and therefore they're 40 map units apart. So I'm going to just kind of arbitrarily go, okay, well, there's A, and there's B. Okay, and then I like to designate the distance. So this is going to be 40 map units. Okay, so now let me get my third gene on there. So my third gene I'm going to put on is the C gene. So let's see, how far apart are the B and C? Oh, they're 20. Okay, so that C gene could be in the middle of A and B, or it could be 20 map units to the right of the B gene. Okay, so let me figure out where is the C gene in relation to the A gene. Well, C and A are also 20 map units apart. So C gene is right here, smack in the middle. That means that this is 20 and this is 20. Okay, so that takes care of that one, and that one, and that one. So now I have the D gene left. So I need to figure out where is the D gene. Well, the distance between C and D is 10. And the distance between B and D is also 10. Okay, well the distance between C and B is 20, so D must be here. And so this distance is 10, and this distance is 10. Okay, so the linear arrangement would be A, C, D, B. Or you could have written it the reverse, B, D, C, A is the same thing. Okay, so when you go to approach linkage maps or trying to figure out the linear order, always start with a line and then start filling it in. Okay, so that was the last genetics problem. Now we just have some of these problems. Okay, so your Are You With Me sheet should be finished. Let's just finish it up with a couple more questions for you. So first question, Labrador Retrievers. Their fur color is controlled by two alleles. Or sorry, let's just say two genes actually. The E gene and the B gene. Any dog with the --EE genotype will be yellow, while dogs that are big B dash, big E dash are black, and little b, big E dash dogs are chocolate. This is an example of, is it epistasis? Is it codominance? Is it incomplete dominance? Or is it linkage? Let me give you a minute there. So hopefully you answered epistasis. We would say that the E gene is epistatic or controls the expression of the B gene. Okay, and in the case of lab fur color, at least one dose of the dominant E is required to have color, either black or brown. Okay, without one dose of the big E gene, which is shown, which is shown here, we end up with yellow. Okay, which means that no color gets deposited. Okay, second question. X-linked recessive traits in humans or in Drosophila, fruit fly, are observed blank. Are they observed more in females? Are they observed more in females than males? Or are they observed in males and females equally? Or are they observed in different distributions depending on the trait? So this takes us back to actually the last video that you watched with X linkage. Do you remember? So hopefully you answered red. X-linked recessive traits like color blindness or hemophilia, they're more common in males than females. And that's because males only get one dose of that X chromosome. So they're hemizygous. Whatever they get, they express, where females have two. And so if they get one, bad recessive allele, they may have one good allele to mask that. And then they would not end up with the disease or the disorder or the condition. Okay, so this is the end of chapter 12, part 2. So you should be finished with your Are You With Me? So you want to go ahead and upload those to Blackboard. And then you want to try the genetics problems. Hopefully you can get those done. And then you want to go ahead and take the quiz for this chapter. okay? And then you're going to have your first exam covering chapters 11 and all of chapter 12, part 1 and part 2. And that'll be next week.